Okay, good afternoon. So my name is Christina Streets. I'll be presenting with Betsy Ballard, the drug device combination products, methodologies for user interface evaluation. Today, our objective is 1, to provide key principles for comparative analyses and understand key comparative analyses definitions. 2, discuss FDA's experience with comparative analyses. 3, explain the human factors research being conducted by the FDA. And then we'll describe the taxonomy and how to implement it. So I'll begin. First, I'd like to start with some key definitions and information regarding generic drug device combination products. Therapeutic equivalence considers whether drug products are pharmaceutically equivalent, bioequivalent, and whether the products are expected to have the same clinical effect and safety profile for the conditions of use specified in the labeling. Therapeutic equivalence is also expected for generic drug device combination products and takes into consideration whether the end user would be able to appropriately administer the drug product without the assistance of a healthcare professional or without receiving additional training prior to use. And that's when the generic product is substituted for the RLD, the reference listed drug. This does not mean that the two products need to be identical, but the differences must be acceptable and allowable under the abbreviated new drug pathway, the ANDA pathway. Excuse me. So how do applicants and how does the FDA determine whether these differences are acceptable? The draft guidance, comparative analyses, and related comparative use human factor studies for drug device combination products, sorry about that, submitted in an ANDA, which was published back in January of 2017, provides applicants with guidance on how to conduct comparative analyses to assess differences in their proposed generic device compared to the RLD. The guidance describes 3 comparative analyses to be conducted. The labeling comparison should be a side-by-side comparison of the proposed labeling compared to the RLD, and for our purposes, focuses on the delivery device constituent part of the labeling, and that would be in the prescribing information and the instructions for use, and then any other related labeling. In the comparative task analysis, the applicant should take apart and analyze all the potential differences in the use process for the proposed generic product when it's compared to the RLD. And then the physical comparison should evaluate the physical design differences of the proposed product to the RLD, and it should include both a visual and tactile examination. To conduct a comparative analysis, it's important to first understand some of the key definitions. When we talk about the user interface, we're considering all of the components with which the end user interacts. This could include the delivery device, this includes the delivery device constituent part, as well as any controls, displays, product labeling, and packaging. The critical task is defined as a user task that when performed incorrectly or not performed at all, would or could cause harm to the patient or user, where harm includes compromised medical care. Identification of critical tasks takes into consideration the indications for the drug product, the users, such as patients, caregivers, or healthcare professionals, and the environment and other conditions that might influence the importance of a particular task. External critical design attributes are those features that directly affect how users perform a critical task that's necessary to administer the drug product. So what have we seen in terms of submitted comparative analyses? Between January 2018 and December of 2023, we received over 1,000 comparative analyses for ANDAs. The majority of the submitted comparative analyses based on the route of administration were injection products. That's followed by ophthalmic products and then oral products. We also see nasal, inhalation, and ODA products, as well as products we've classified here as topical, vaginal, and rectal products combined. What are the possible outcomes when conducting comparative analyses? The draft guidance mentioned earlier provides detailed information regarding the 3 possible outcomes. 1, no design difference. 2, minor design difference. And 3, other design difference. No design difference is exactly what it means. There's no difference between the proposed product and the RLD. Minor difference means that there is a difference in the user interface of the proposed product compared to the RLD, but this difference is not expected to affect an external critical design attribute, which I remind you is a feature that directly affects how a user performs a critical task. And finally, other design differences. These are differences in the user interface design that may impact an external critical design attribute and therefore may impact a critical task. And the important part there is that it may impact. There we go. So what does it mean when a product's determined to have an other design difference? How does it affect approvability as an ANDA? If it's determined that the product has an other design difference, it still may be approved as an ANDA, but it may require additional evaluation. We need to consider how the design difference could impact any errors, question whether it changes the clinical effect or safety profile of the proposed product compared to the RLD. And we take all of this in consideration of the RLD as well as a specific ANDA. We look at the context of use of the product, such as the end user population, the environment of use, the urgency use, and also the frequency of use. Sorry about that. So how often do we see other design differences that are unacceptable? Of the over 1,000 comparative analyses I mentioned previously, there were 83 applications that had other design differences that were unacceptable. This is approximately 10% of comparative analyses. This slide does not show other design differences that we deemed acceptable, though. So these are ones in which the other design difference was unacceptable. The other thing that we'd like to make note here is that often throughout the review cycle, we are able to make recommendations so that these other differences are, made to be minor differences or no difference potentially. And so we only send a complete response letter if we're unable to rectify the issue during that cycle. The majority of products that had other unacceptable differences were injection products, accounting for over half of the applications. Oral products made up a quarter of the applications, and then ophthalmic products and those products under the topical, vaginal, rectal were the remaining applications. When we identify these unacceptable design differences and issue a deficiency, we recommend that the applicant consider modifying the design of the user interface to minimize any differences with the RLD. If the applicant does not intend to change their design, then we recommend that the applicant submit additional data to support or justify that the other design difference will not alter the risk profile of the proposed product when it's substituted for the RLD. Examples could include in vitro studies or a comparative use human factor study. The comparative use human factor study has been an area of research interest for the FDA, and I'll pass on the presentation to Dr. Ballard to discuss further. Dr. Ballard?
So just to get an idea, a sense of the audience, show of hands for those in the audience, and I think you can vote online. How many of you have submitted and/or conducted a comparative use human factor study? So yes, you have, no, you haven't, or you have no idea what I'm talking about. So how many have submitted? 1, 2, 3. So how many have not? The vast majority, I'm assuming, have not. Okay. That's kind of what we suspected, because this seems to be a pain point when you have to consider doing a comparative use human factor study because you have an other design difference. This has been considered a pain point, and so it's one of the reasons why the research program was started. So this is just a quick timeline because I wanted to show sort of the relative the recency of this whole area of research that's the FDA is conducting. The draft, as Dr. Streets said, went out in 2017. In 2020, at the start of COVID, OGD stood up what's considered the device evaluation team. And we work in the pre-ANDA space to advise industry and companies on design differences for their proposed DDCP and help them work through some of the issues and how to obtain data that will allow them to then go on for approval. So as part of that, in 2021, the request went out for proposals to study the comparative use human factors arena. And that, while some of you in here are probably familiar with the traditional human factor studies like the validation studies or usability studies, the comparative study is different because it requires you to compare your product to a reference product rather than comparing it amongst its using your device design and having users evaluate it and make iterations and design differences based on that. So it's a little bit different. 2 grants were awarded that year. One was entitled User Interface Designs for Generic and Reference Listed Drugs, and that went to Battelle Centers, the Public Health Research and Evaluation. And the other grant that went out was for the development of a taxonomy to help identify these things, and that was granted to the University of Detroit. So the first grant, the one that went to Battelle for the user interface designs for generic and versus RLD combination products, had 2 aims. It wanted to develop enhanced methods for threshold analysis and categorization of user interface differences. And the second aim was to try and establish effective methods to assess these other design differences. Unfortunately, that grant terminated early and there are really no outcomes to report. The second grant, the development of the taxonomy, from the University of Detroit, had 3 aims. The first one was to develop a body of knowledge of key stakeholder perspectives and existing strategies that people were using to assess the user interface design differences. The second aim was then to use that information and build on it and develop a visual taxonomy to assess the user interface for combination products. And finally, to use that taxonomy and actually apply it in a comparative process using a reference drug and a generic drug. So what is a taxonomy? When I took this project on, I had no idea what they were talking about. But it's a method for organizing specific concepts and creating a common vocabulary for those concepts, so that when we talk about something, everybody understands what we're saying. And the key thing that the University of Detroit collaborators wanted to do was link that design feature to a specific task and risks that are associated with that task. Okay. So what did they actually accomplish? They did accomplish aim 1. The interviews were completed with the key stakeholders. They interviewed human factors engineers, CEOs, and a variety of people, and they performed a literature search to see what was available in the public arena on comparative use human factor studies rather than the traditional human factor studies, and prepared a publication which is currently undergoing peer review. For aim 2, they did develop a taxonomy, and that's what I'm going to talk about in the subsequent slides. And they validated this taxonomy in a small inter-rater reliability study. They are developing a case report using their taxonomy and fabricated data to show how this taxonomy can be used to determine whether your design differences are other or minor. Aim 3, unfortunately, which was applying this in a broader concept, was not completed. So what is the taxonomy? So it's they started with the combination product categories, and they used a 21 CRF 3.2E, and they used that to help define what a combination product was. And then after talking with the agency, they were able to prioritize which products they would look at in more depth. They then you looked at each of those devices and developed task analysis for each device that was very detailed. And they also looked at developing use error analysis for each of those tasks and for each of those devices. And they linked that to the URAA. They then linked that risk and the task back to a specific design feature and came up with a taxonomy. And what they started with, guys, it's not advanced. Okay. What they started with was basically look like this. And, you know, it kind of reminds you of the elemental chart in chemistry. They basically identified two major areas. One was labeling and one was user interface. They then went down and broke each of those down into certain specific categories, and then subcategories and descriptors. So the further down you get on the table, the more specific you're looking at. So on the and this is sort of a breakdown, and it probably shows a little bit better one of those particular categories and how you can trace it down. When you're looking at your device, you would start at the descriptor and work your way back on up through the thing. But they categorize each design attribute by color, size, shape, texture, and material. So very much like our guidance says, you want to look at the shape, size, and orientation of each product. And they looked at the literature and found what were the risks associated with each of those particular design features. They realized that shape and form, so a minor difference they decided would be a change within a subcategory, whereas another difference would be a change in a critical design feature, so that it would be the difference would be higher up or in different categories altogether. So this was obviously a very cumbersome way of doing it, and they developed a spreadsheet that incorporated each of these tasks and walks the user through a series of questions that delve further into the hierarchy and allow you to classify it. So it looks something like this. And you can see this is for a blood pressure cuff. And so you can see that they have it broken down by the group. The tasks are all there, and each one is assigned a group. And it's color coded, so it's very easy to look at, and you can just get a very quick visual as to where your differences are and where you might have to concentrate on looking at things further. They ultimately wanted to use the results of this hierarchy, this spreadsheet, and you would do one for your RLD and one for your test product, and you could quickly identify where the differences were. And then the goal of the project was then to take those differences and assign each thing a risk and come up with like sort of a relative risk value and produce what they are calling a determination report. And so in order to be a minor design difference, that it must link to a non-critical task. It cannot add any new or increased potential for harm. It cannot add or subtract or eliminate a task. It can't change the action required to complete that task. And it must fall within the same descriptor within the taxonomy. If a design feature does not fit those, then there's potential that it would be considered another design difference. And they propose that because this could be, because each of the ultimate value of this would be the risk that would be associated with this, you could use this to help you justify why your design differences may or not be acceptable for an ANDA submission. But at this point in time, the acceptance of this report or any report such as this in an ANDA submission is a bit premature because none of this has been finalized or validated. Where do they see where do our collaborators see the future going? Well, clearly, even a spreadsheet is not the easiest to work with, and so a web-based taxonomy tool would be ideal. And they want to further take the step one step further and use those results to help you identify minor versus other. And that'll be a risk-based task and task-oriented assignation. They need to apply the taxonomy in a larger study using a variety of participants. So not just human factors engineers or engineering students, but also industry reps and potentially even regulators to see in an interrelated reliability, how often do we agree on the classifications. And that will allow them to continue revising and updating the taxonomy tool. So that project terminated in June of this year. And so what ongoing research does the FDA currently have in the human factor space? And what we have is an IDIQ, or an indefinite delivery, indefinite quantity And the goal, because of all the feedback we've been getting from the agencies about trying to do these comparative use human factor studies and all the problems that are associated with them, they decided that the task one for this IDIQ would be to actually conduct a human factor study the way that our guidance suggests that we do, and following the recommendations that we give to industry, so that we could identify where exactly the problems are, what are the issues, and how can you move past them and resolve those issues, so that information can be published and be available to industry to help you do a better job conducting a human factors potential study. And the other interesting thing about this would be, once we have this data, then we could take it and look at evaluating with potentially different statistical methodologies. And that's very exciting at this point. So in summary, the taxonomy, it can be a powerful tool because it provides a common language. It can assess, the way this one was designed, it can assess the level of risk associated with the design differences, but it needs to be implemented in a larger study of the comparative process. The Office of Generic Drugs continues to fund human factors research. The IDIQ went out in March of last year. The devices are being selected, and the comparative use human factors study protocol is being developed. But what I really want to draw your attention to is the broad agency announcement that will go out in November of this year for FY 2025 to encourage anybody who might be interested in coming into this comparative use space to go to that webcast, which is, I think, November 7th, and certainly submit your proposals for consideration. And with that, we'll go on to the challenge question. Which of the following is true regarding other design differences? A comparative use human factor study is always required. The design difference may impact a critical task. They preclude approval under an abbreviated new drug application, or the applicant must modify the design difference to minimize it. And so anybody want to, do we have the results from the poll? They're not coming up, so that's what I'm asking. Okay, the answer is the design differences may impact a critical task. And challenge question number 2. Which of the following are current research projects that the FDA is conducting? The taxonomy development, the user interface design for generic versus combination products, a comparative use human factor study, or there's no current research in this space? And the answer for that is we are in the process of conducting a human factor study. And with that, I'm going to turn it over to Dr. Wang, who's going to present her topic.